All right, thank you very much, Philippa, and thank you, Orox, for this kind invitation as well. It's been a fantastic session, knowing about new technologies that are coming up and various other talks that are very technologically oriented. I'm going to focus mostly on a very scientific aspect that we, take part in our lab, while showcasing some of the technologies that we use for this purpose. But before going there, I want to emphasize on the main philosophy that we follow in a lab. Typically, you would find in any biological study that protein X regulates process Y. And we sort of try to understand what that word means. What does regulate mean? Because it doesn't define anything very, very precisely. It just implies that that protein probably plays some sort of a regulatory, by increasing or decreasing the process, etc. Right? So I kind of would like to point you towards this nice review by Eran Pichetsky, where he argues whether the concept of regulation is overused. In our lab, what we like to do is when we talk about molecules, when we talk about protein interactions, protein membrane interactions, what we want to understand is what are these interactions, how do these interactions matter, and how do emergent properties arise out of this, and why do these interactions exist? And all these answers we seek to get by performing quantitative microscopy. So we want to measure everything. We want to measure everything quantitatively, use computational image analysis, and then perhaps at the end employ simulations or mathematical modeling to better describe these biological processes. And so one of the framework that we use to study these kind of processes is noise, right? So the biology is full of noise. There are various factors which gives rise to noise in living systems. The most well-known things are in gene expression patterns, where you can see that a lot of feedback loops exist to suppress noise. Sometimes molecular interactions use noise as a means to have sort of a play within the space in which they can interact and give rise to different properties. So what we want to ask here is, if you take the case of membrane-bound organelles, essentially endosomes, Golgi, mitochondria, when there is exchange of materials between them, what gives rise to noise, and how does the molecular interactions help to suppress those things, right? So before going there, I'd like to introduce briefly the overview of what endosomal trafficking is, at least the part that we study in our lab. So when you look at the plasma membrane, you can have various endocytosis processes. It can be through clathrin-mediated endocytosis or clathrin-independent endocytosis that gives rise to vesicles. These intracellular vesicles that are about 100 nanometer undergo clathrin uncoating in the case of clathrin-mediated endocytosis, and successively fuse to form larger endosomes. At that stage, they are known as early endosomes or very early endosomes, which can undergo multiple levels of tubulations to give rise to recycling endosomes. There are various kinds of recycling endosomes, fast and slow. The leftover material is successfully carried on to something that's called late endosomes, which then goes on to become multivesicular bodies where degradation takes place. You can also directly go from early endosomes onto Golgi. Also, Golgi also releases vesicles that go out onto the plasma membrane to fuse and perform exocytosis. Now, in this whole process, there's a lot of other events that take place simultaneously, and that build up this whole network, right? So, namely, these events are proteins that define the membrane identity. For example, early endosomes are defined by the presence of RAP5, late endosomes are defined by the presence of RAP7, etc. Through these endosomal populations, there's a lot of other chemistry that happens where the phosphoinositide species are converted from one form to another through a lot of other enzymes. Simultaneously, while all this is happening, these endosomes are pulled and pushed by motor proteins called dynein and kinesin. That dynein takes these endosomes towards the center of the cell. The kinesin takes the endosomes towards the periphery of the cell. And then there are a lot of adapter proteins that interact with specific receptors or specific cargos that also have on-off binding rates. Right? cargo sorting, wherein membrane can undergo tubulation and the cargo can go from the main body of the vesicle into the tubules, is also a very noisy process. So all these processes that build up the endosomal trafficking are very, very noisy. So we would like to understand, how does the cell, despite of all this noise that is present in the system, carry out its trafficking process very, very robustly, right? So if you look at imaging approaches to study these, the process of endocytosis and exocytosis has been studied very well, simply because this can be accessed very nicely through total internal reflection fluorescence microscopy, that you can only excite the bottom 100 nanometer thin layer of the surface of the cell, and therefore you can visualize these events that take place with very high signal to noise ratio, as well as very high resolution if you're using a high resolution, high NA objective, right? So in this case, you can see an example of endocytosis of clathrin coated pits and exocytosis of sugar toxin simultaneously. However, when you want to image the processes that happen deeper inside the cell, you need to have a couple of parameters taken care of. And these parameters are spatial resolutions. These endosomes are of the order of submicron. You need good time resolution because these are pulled and pushed by motor proteins that move on an average with a velocity of a micrometer per second. And you need volumetric speeds because these things are moving in 3D, and therefore you would like to have a good volumetric speed in imaging. Moreover, the way you excite the sample needs to be very, very precisely matched up with what you're imaging, because otherwise out of plane excitations can give rise to phototoxicity and photobleaching that decreases the longevity of your experiment. So here's another case for why we need to do fast and good live cell imaging. So think of it as a single cell, and these are timestamps of the single cell. You start with, let's say, 3 green endosomes, and slowly these green endosomes start turning into magenta. So here is an endosome that is turning into magenta. This endosome has already turned into magenta, while there are new endosomes generated here named 4 and 5. And so if you follow this process, and if you just make a kymograph of each of these endosomes, you would see something like this. But when you fix the cells and just look at them, you would simply say, well, there are 2 green endosomes, 2 magenta endosomes, and 2 endosomes that have green and magenta on them. This really does not give you the full picture. This does not really tell you how the endosomes are moving and what are the rates of conversions, what are the overall rates of number of endosomes that are generated with respect to time. And so if you want to do all this, if you want to get this complete picture of this dynamic system, you need to be able to do live cell imaging. So as I said, you could perform TURF to get a picture of what's happening at the bottom 100 nanometer membrane of the cell. If you do confocal imaging, you can scan in this manner. You get a single plane with reasonable resolution, yet you get these out of focus excitations that results in substantial photobleaching. And when you do 3D imaging, it's rather slow, and you would get a tremendous amount of photobleaching. The speed is necessary because if you want to characterize endosomal motilities and track them while they're moving, if you're not capturing enough number of frames in between 2 positions, you would not be able to say whether this endosome after a certain time is this one or that one, because you're not able to link them properly. While if you have continuous positions of this endosome, you would be able to link them more faithfully and get a much better picture of what exactly is happening there, right? And so the approach we've taken is that of light sheet. In this modality, what we have is a light sheet that slices through the cell, and we get multiple of the slices, put them together, and you get a volume. You repeat them again and again, and you get a time-lapse volumetric movie, right? So one of the tricks that we use is to use a very thin light sheet, and this thin light sheet, the method of generation comes from Eric Betsick's lab, and we use this light sheet called the Lattice light sheet, where you have multiple vessel beams that coherently interfere to give rise to this kind of pattern. And if you deter this pattern fast enough, you get something that looks like a thin light sheet. So the reason why this is very much appropriate for this sort of study is because when you look at the thickness of the excitation, that's about the order of the wavelength. and that is very well matched up with the field of depth of field of the detection objective. So in this case, when you look at the field that is excited by the excitation objective, it's exactly that planar field that is imaged by the detection objective, and there are no autofocus light that leads to substantial photobleaching. Moreover, you could scan the cells through this sheet of light very, very rapidly and get fast movies. So here's an example of two movies with orthogonal projections. This one was one of my first movies that I took with Gokul at Kirshausen lab when I was first exposed to this microscope. And this was when I came to UNSW and established my own lab looking at endosomes. And so coming back to the scientific questions that we are after what sets the rates of maturations of this endosome from one population to other, and what are the mechanisms that exist to maintain these rates of maturation. So we're simply going to look at three proteins here in this talk. One is APPL1 that binds to PI3P. The other one is EEA1, which has got a special structure, which I'll explain as we go through the talk, and RAP5, which generally marks the very early endosomes and the early endosomes. Through this process, there is a phosphoinositide conversion that happens. So PI3P gets converted into PI3-4P2 by means of INPP4A. And so this process has been looked at it earlier in the literature. So this is a paper from Pietro Di Camilli's lab, where they've imaged APPL1 to EEA1 conversion. What is striking is that you could see the same endosome go from one form to the other. However, this paper reports only a very few number of events and does not report whole cell level events in the manner that you can characterize the overall rates of these conversions. And so therefore, we decided to look closer into this matter and ask what sets the rate of these endosomal maturations. And so one of the first things that we observed was when you look at this whole cell image of APPL1 and EEA1, there's a lot of inter-endosomal collisions, right? And we can follow, I'm not going to go very deep into the image analysis methodologies that we have performed and are working upon, but I'll generally tell you the results because of limitations of the time. So if you look at these endosomes, they all collide with each other, and there's a lot of heterogeneous collisions. So we quantified the number of collisions that result in conversions and the number of direct conversions. So for every cell, if you look at the frequency of the events in the 15 minutes of the volumes we take, we see that a lot of events, a lot of conversion events do arise out of collisions between the endosomes. And so to substantially validate that, we inhibited dynein using ciliobrevin, or we mildly depolymerized microtubules using nocodazole, and we saw that the number of conversions over 10 minutes per cell really drastically decreased. So that means that there's this high conversion zone that we observe in the cell where endosomes are always colliding with each other before they undergo conversion. And as soon as they undergo conversion, you can see that the green endosomes turn into magenta endosomes. And so then we started looking at what is the structure of this protein and why are these collisions important. So EEA1, the protein in magenta here, is a protein that's 200 nanometer long. So it's almost the size of the endosome itself. And it's got two domains, the N-terminal domain and the C-terminal domain. Also, EEA1 is a head-to-head dimer, so this is bivalent at both sides. And the specialty about these domains are both of these domains bind to RAP5, except that the C-terminal domain can also bind to PI3-4P2. And so I'll come more into that later in the talk. So here's what I mean by what's happening in the collisions and the fact that collisions are significant for conversions. So here's a zoomed out view of APPL1 endosome that will go and collide with this EEA1 endosomes. And these are just tracked, the trajectories of these endosomes that are tracked using manual annotations. And these are the intensity traces of APPL1 endosome that will undergo conversion versus the EEA1 endosome that is the parent endosome. So as you see, these endosomes starts as an APPL1 endosome, comes and collides with this EEA1 endosome. And as soon as it collides, you can see that it starts to turn into an EEA1 positive one. And subsequently, it goes and merges with the parent EEA1 endosome. Right? And if you look at the intensity traces, you see that the same endosome, which has the APPL1 signal, does not have much of an EEA1 signal, but this is the point where the collision takes place. And after the collision, you can see a lot of fluctuations arise before it fuses with the parent endosome. So this is the point where it can be termed as a converted endosome. And so if you look closely at the knockatazole treated cells, you can see that the endosomes do convert, but they don't completely commit to conversion. They go back and forth a lot, right? Whereas if you look at endosomes that have no motor protein inhibited, you can see that as soon as they collide, they start converting and fusing with the parent endosome. Right? This tells us a couple of things. There's something about the collision and fusion that is making sure that the conversions go forward and complete the reaction. And so we looked closely at what is the organization of EEA1 on this endosome. So we performed 3D super resolution localization microscopy, and we saw that these EEA1 are clustered and they form domains on these endosomes. So here's a 3D rendering of a single endosome, and you can see that the EEA1 does not cover the entire surface of the endosome, but rather covers a small domain on the endosome. You can also perform live, and you can see that these endosomes look like they form very strong clusters along the circumference, and they also fluctuate a lot. If you look at the co-localization between APPL1 and EEA1 on the same endosome, we find that they are counter-clustered, so they don't co-localize at all, but the APPL1 is present where EEA1 is not, and vice versa. Now, if you look at the time characteristics of the EEA1 endosome, which is undergoing a conversion, you can see that the APPL1 signal decreases, but the EEA1 signal increases, but it increases in a manner which points towards clustering and very strange oscillatory behavior. And so what we figured out by imaging, essentially volumetric live cell imaging and super resolution, is that collision between endosomes resulting in direct fusion or not are important for conversions. EEA1 is clustered on the endosomes and shows large oscillations. EEA1 is counter-clustered to APPL1, suggesting that there's a negative feedback in the reaction scheme. Right? So, as I said, EEA1 has two domains, N and C terminal domains, and you have the N terminal and C terminal both interacting with the RAP5, and the C terminal interacts with the PI3P. So we wanted to know, in the journey of this endosomal maturation, is there preference for one format versus the other, or do both of these organizations exist, or is there a precedence in the orientation as the endosome matures? So we performed fluorescence lifetime, where the N terminal of EEA1 is labeled with a donor, and the RAP5 is labeled with an acceptor. So in case of N-terminal binding, you would expect that the FRET takes place, and you would have decreased donor lifetime. In the case where the C-terminal is bound to the endosome, you would have no FRET, and therefore the donor lifetime would not decrease and would exhibit a longer lifetime. So in our color schemes, the shorter lifetime will be depicted by blue, and the longer lifetime will be depicted by green, and anything in between would be depicted by cyan. So here's an example of the movie. Here you have RAP5 signals, and here are EEA1 positive endosomes. And you can see here the lifetime, measured lifetimes of EEA1 to RAP5 FRET in blue or green. Right? So you can see there are different kinds of populations. So here's an overview of the area. You can see that a lot of endosomes have blue, are blue in color, meaning that there is FRET taking place, which means that the N-terminal is the one that interacts first with the vesicles. And as the vesicles mature, they and grow larger, you see more green, which means that there is no FRET taking place, and therefore it's bound mostly through the C-terminal. We also see many other rich features while observing fusion of these endosomes, etc., which we not performed a very detailed analysis and are not ready to talk about. What is clear from this experiment is that the orientation precedence is the following. The N-terminal interacts with RAP5 before the C-terminal can interact with the RAP5 and PI3P. So what does this mean in the scheme of the entire maturation process? So you have Pi3-4P2 that converts into Pi3P, and you got N-terminal that interacts with Rab5, C-terminal that interacts with Rab5 and 5. You have APPL1, the protein that will interact with Pi3-4P2 and Rab5, and you got Rab5 itself that interacts with both of them and is membrane localized, right? So these are the various interactions on molecular players that are involved. Of course, these interactions are not very linear. So here's the overall scheme of the interactions taking place. What we've observed is that this is the first interaction that takes place, and this is the second interaction that takes place. And in the scheme of these things, Pi3-4P2 is getting converted into Pi3P. And so we started modeling these processes to sort of visualize whether these collisions, clustering, and the sort of orientational precedence can result in a robust endosomal conversion. So here's a schematic depiction of, sorry. Sorry about that. This movie is causing trouble. So this is a simulation of the whole process, that the reaction scheme that I just described. I think there's a problem with playing the movie, so I'm not going to play the movie. But here you can see that these are experimentally measured EEA1 intensities of a converting endosome. And these are the simulated versions of EEA1 oscillations. And so we are pretty much able to recapitulate a Pi3-4P to Pi3P conversion in a Rab5 positive endosomes with APPL1 and EEA1 intensity or population fluctuations taking place in case of simulations. So if this movie played, you would see that the APPL slowly comes down, and so does IMPP4A, while EEA1 keeps increasing and overpowers everything else. So in short, what these experiments suggest is that without collisions, there is a propensity for these reactions to take place on the endosomes. However, they don't proceed very well to completion in time, and so they can have a lot of back and forth movements. When you have these collisions that are mediated by motor protein motions, clustering, and intra-endosomal conversions, every time there is a collision, you would have an ensured APPL1 endosome converting into an EEA1 positive endosome. And that means if you look at the population level of all the endosomes in a cell, every collision ensures that the endpoint of a mature endosome is met within a very short distribution of time scale. Or in other words, the first passage time of these endosomal conversions are maintained very robustly through collisions and a clustering of EEA1. And so in detail, what we've shown using fluorescence lifetime is the mapping of this orientational precedence of EEA1. So here's a mature endosome that has Pi3P on it, C-terminally bound endosome, that in the FRET experiment would appear green. And so these endosomes collide with incoming new endosomes that have mostly N-terminally bound EEA1 on them. And this results in an endosome that has both the populations, which then matures, right? So this kind of cycle keeps happening, and this essentially tells us that the EEA1 acts as an early endosomal acrobat, where the structure, this exquisite structure of C-terminal and N-terminal is designed to execute these collision-mediated conversion behaviors. Right. With that, I would like to thank the actual people who did this work. So Harry, who's probably in the audience, carried most of this work forward, including the Lattice light sheet imaging. Ullas was also instrumental in carrying out the Lattice light sheet imaging. Abhishek and Hetvi analyzed a lot of these data. Hetvi analyzed a lot of this data manually as well. We collaborated a lot with Shree's group, which is a group that specializes in stochastic physics and image analysis. Charlie wrote a lot of code, and we are developing a lot of codes for analyzing endosomes of Lattice light sheet movies. Kunal performed the simulations for our project. And I'd like to thank the funding bodies. And with that, I'm happy to take any questions, while I can advertise a bit for our latest paper where we have combined optogenetics with Lattice light sheet microscopy.
Thank you very much indeed, Senthil. You're finished early, so we have plenty of time for questions. So please please would people put their questions in the chat box for Senthil. Obviously, he'll stay online to answer any questions afterwards. So we have one question coming from Bob Hartley. He says, does the cargo affect the maturation?
Sure. So cargo does affect the maturation. I'm just going to go. Yeah. All right. So we've not tested a whole lot of cargo, but we have tested EGF, which is a cargo that goes through the endosomal cycle and ends up in the lysosome. And so this is a paper that we published this year, where we show that when we add EGF to the cells, it triggers calcium waves that results in APPL1 being recruited to all the EGF pits, and results in cohort trafficking of all the EGF towards the periphery of the cell. So it certainly, cargo plays a role in regulating and perturbing the steady state of endosomal maturation. And we see that it should affect the rates of endosomal conversions as well.
Okay. Which software are you using for your visualizations? They're beautiful. They're a little bit jumpy on the screen, but I can see that they're really nice.
Oh, sorry, I didn't realize they're jumpy on.
That's
on
okay.
screen.
We can get the gist of it. It's showing every frame, every few frames. I think.
So when I started the lab is not very old. It's about 4 years old. So when we started the lab, we didn't know what to use. The Lattice light sheet was new. So we started using a lot of software. We've mostly used Imaris to visualize. Slowly, we are trying to use Avia as well. Abhishek from my lab is also developing his own visualizer based on Napari, which is a Python-based visualization library. So yeah, we're going after a bunch of things. We're also using SciGlass, which is fantastic to observe membrane ruffles, for example, using virtual reality.
Okay. Oh, that'd be really cool. So you can actually manipulate it while you're seeing it real time. That would be really good. Yeah, I must say I've played with both Imaris and Avia, and they're both really very powerful software packages. So I know that Patrice from Avia is going to be giving a presentation tomorrow. So I look forward to that. Do we have any other questions for Senthil at this time? And if not, is Dr. Bat ready to start? Because if he is, then we can run a little bit early. Thank you so much for that talk, Senthil. I'm afraid I can't ask very many questions about the biology, being a chemist in training. Bob has asked another question. He said, have you ever used TURF/HILO, and how does the Lattice light sheet compare?
Sure. I mean, TURF is going to only excite the bottom 100 nanometers, so you're not going to ever track the endosomes in the three-dimensional volume of the cell. You could use HILO. I'm not very convinced that HILO is a laminated excitation profile. I think it's more of the weird scattered beam and the cross section of the very thin depth of focus of a high resolution objective. You could perhaps put in a cylindrical lens or model the PSF in a way to track these punctate structures, but you do lose a lot of get a lot of aberrations when your endosomes grow larger and larger in size. And so therefore we prefer to use Lattice light sheet, where it's actually the excitation profile is very clean. We know exactly what we're imaging and the cross sections of excitation versus collection of the emission, etc. Okay.
Thank you. I hope that answers your question, Bob. Obviously, Senthil will be around in the background. So if you have more questions for him, then feel free to keep them in the chat folder. So thank you so much, Senthil. I will be speaking to you soon about our recent discussions anyway. So let's chat soon.
Yep. Okay. No worries. Thank you, Philippa.
Thank you very much.